Uh, Hi everyone. Today I am going to give you in detail about animal cell culture. First of all, I want to let you know what is normally cell culture. Is the process of growing either animal or plant cell outside of the body in vitro. That is called cell culture. Normally, we culture animal cells. Uh, so the process of growing animal cell outside or in vitro condition is called animal cell culture in controlled laboratory conditions. So during the cultivation or growing of animal cells, we have to maintain the sterile container like a flask, bioreactor, even a nutrients rich culture medium which is essential for the growth of animal cells. And we also need to supply the supplements like vitamin, amino acids, salts, and other growth factor which support the growth of animal cells. Basic condition for growth of animal cells normally requires like a sterile environment, as I told. If we are going like bacteria, fungi, we need a sterile environment. As same for the animal cell, it requires the same kinds of environment, and we need nutrient medium. On that case, while doing animal cell culture, normally we use double co modified agar media, which is also known RPMI 1640, and we supply 5% of carbon dioxide atmosphere in the carbon dioxide incubator. pH is normally um, maintained between 7.2 to 7.4. There are so many kinds of animals cell culture. Among them, I'm giving some of the examples and their characteristics and application. Like first, HeLa cells. HeLa cells is cells that derive from the cervical cancer cells of patients named Renita Lax 1951. And the characteristic of this cell is this cell grow very fast and has robust characteristics. It can survive a long period in culture as well as is easy to maintain in the laboratory conditions. So it is in choice for the culture. Application basically use this HeLa cells for cancer research, drug testing, and genetic research. So to test whether the HeLa cells has shown any kind of effect or not by virus, researchers have done a kinds of experiment on which infected HeLa cell with a virus to study how virus enter inside the cell and replicates in human cells like that. Another examples of cell culture is CH cell, co-cell, which is also a Chinese hamster ovary cell. And the characteristics of these CHO cells is they grow in suspension or like in attached culture media. They can easily genetically modified so that they are choice best choice for like large amount of protein as well. If we have to understand if we wish to understand the applications, it is used for the biopharmaceutical protein production, monoclonal antibody production, biotechnology and pharmaceutical industry. Examples like many therapeutic antibodies used to treat diseases like cancer on using CHO cells grown in large bioreactor. And another cells which is popular among other cells, vero cells that is derived from the kidney epithelia cell of African green monkey. The characteristics of this cell is they are stable and easy to maintain, and also they are sensitive to many viruses and used basically in the virology research. Why it is important in the field of research study? The reason behind is like they are able to support for the vaccine production, virus isolation, and toxicity testing. One example, for this virus cell is they are used to produce vaccine for diseases like rabies and folios. And the other kinds of cells is a H 293 cells. They are originally derived from the human embryonic kidney cells. So they have a characteristics like they can take foreign DNA and highly efficient as well, and they are ideal for the gene expression. That's why they have applications in recombinant protein production, gene therapy, and cell signaling studies. So scientists insert such kinds of gene in hacker 293 cells and produce specific human protein for research. Uh, some examples of animal cell culture experiment to see like the effect of a drug on cancer cells. What do we have to do? Like in that case, we have to follow some of these steps. Like first, we have to grow HeLa cells in culture flask and maintaining nutrient media. Yes, nutrients, so that then we could able to incubate cells at the 37 degrees centigrade with a 5% of carbon dioxide supply, and we will do different concentration of a drug testing and observe whether the cell are died or live under a microscope, and will determine the result. This is the overall process of animal cell culture. Thank you so much